Yesterday's lesson on last week's lesson we tried to discuss about the basic principles of photosynthesis. At there we have discussed all the important compartments. First we learned uh, about the chloroplast and the chlorophyll molecules and how the excitation happens, and after that we learned about the light reaction, after that the dark reaction or Calvin cycle. So today we are to do C4 plants, an adaptation to overcome the process called photorespiration. That is we are to do to complete about photosynthesis. Before that, as this recording is passed to other students as well, I am going to revise very shortly what we have learned until now. Okay, I will draw some diagrams and then I will revise some. I will revise very quickly and very briefly the things you have learned. Okay. For this I will use some diagrams, so and I'll send these to your WhatsApp group. And first look at the board, and if you have any connection issue or anything, please let me know. First, for the exam purpose, this revision is very important, as I think for the first term test. First we learned about the importance of photosynthesis, and then we have learned that though there is an equation for photosynthesis like CO2 plus H2O with the help of solar energy produces glucose plus oxygen. That is what happens. But I have taught you that this happens in two separate steps. The steps would be the light reaction and the dark reaction, or light reaction and then the Calvin cycle. First, in the light reaction, what happens? Oxygen produces. After that, from the dark reaction or the Calvin cycle, the rest of the equation is completed. So for this I taught you about a mechanism. I taught you about several basic principles. I am not going to discuss them again, but look at this diagram. What happened to a chlorophyll molecule? You have learned that. What happens to a chlorophyll molecule when the light energy comes? When the light energy comes, what happened to a chlorophyll molecule? That's the thing we need to discuss. Okay. This is the chlorophyll molecule. This is the chlorophyll molecule, and when the light energy comes, what happens? The chlorophyll molecule is stimulated. This happens to all of us. When a problem comes, we become aggressive. When someone scolds you, you become aggressive. So, after this excitation, after this stimulation by the light energy, the chlorophyll molecules are stimulated, and what happens? They are excited, and what they do is they release the thing which has attached very lightly to them. What did I say? They release their outermost elements, outermost things like electrons. They release this energy because light energy comes, these are excited, and what happens? The electrons in their outer energy level, what happens? They are excited and goes to an upper energy level. this went directly straight, but directly straight this went to an upper stage of higher energy level, but this cannot come back as quickly as that. Okay, this cannot come back as quickly as that. What happens? This need to go through a series of electron receptors, releasing a little energy here and there and comes back to the normal energy level. This goes through various electron receptors. After that finally it comes back to this original energy level. So this is the basic principle which needs to be understood when we are learning about photosynthesis. I'll recap the equations, I'll recap the light reaction very quickly. දැන් බලන්න මම එදා ඔයාලට කියලා දුන්න එකේ හරිද මුලින්ම මම ගායි chart එක මම අර මුලින්ම basic principles ටිකක් Diani was there I think. Please if you can send us the diagrams. මම basic principles කියලා දුන්න ටික ඔයා පස්සේ class එක ඉවර වෙලා උඩ chat එකට දාන්න එක ෆොටෝ එකක් ගහලා මම ඉතින් අඳින්න කිව්වා මම ඒකේ මේ ෆොටෝ එකක් ගත්තේ නැහැ. ඒක ඒ බේසික් ප්‍රින්සිපල්ස් ටික මුලින්ම පොඩ්ඩක් කියවලා රිසිස්ටර් හතරක් එවමනේ ඊට පස්සේ තමයි අපි light reaction එක කියන්නේ ඉගෙන ගන්න යන්නේ. So in light reaction what happens? Basic principle එකක් තමයි අපි දැක්කේ chlorophyll excite වෙන එක. After that in light reaction where does the light reaction happens කියලා question එකක් අහනවා. ඔයගොල්ලෝ දෙන්න ඕනේ මොකද structured paper එකේ they are asking where does the light reaction happens. Where does the light reaction happens? We are talking about the plant cells here, not the prokaryotic cyanobacteria. We know දන්නවනේ photosynthesis කරන්න පුළුවන් organisms ඉන්නවා. There are several organisms like cyanobacteria, algae and eukaryotic plants. But the thing is, now here we are talking about the eukaryotic plant cells. So we can say in eukaryotic plant cells, in chloroplasts, in granas, in thylakoid membranes, in photosystem. That is the complete answer. So, what is a photosystem? The photosystem are collection of chlorophylls, collection of photosynthetic pigments in a structured way. හරි ලස්සන්ට මේ හැදිලා තියෙන හොඳ ස්ට්‍රක්චර් එකකට හැදිලා තියෙන හරි chlorophyll pigments එකතු වෙලා හැදිලා තියෙන ස්ට්‍රක්චර් එකක් තමයි මේ photosystem එක කියලා කියන්නේ. So in a photosystem there are two things. So in a photosystem there are two parts. The outermost part is the light harvesting system. The innermost part is the reaction center complex. Light harvesting system, reaction center complex. So in the reaction center complex, I told you that there are special chlorophyll A molecules. There is a pair of special chlorophyll A molecule. In one kind of photosystem, we name it PS2. In one kind of photosystem, we name it PS2. These special, these pair of special chlorophyll molecules, we call them P680 chlorophylls. Not only that, in a photosystem there is a primary electron receptor. This is the primary electron receptor. Other photosystem is the other photosystem is the PS1 photosystem. In a PS1 photosystem also, what happens? Here there are two things, two areas actually. There is a pair of special chlorophyll A molecules. Pair of special chlorophyll A molecules. In PS1 photosystem we call them P700 chlorophyll molecules. And also there is a primary electron receptor. So, in light harvesting system, what will they have? They will have photosynthetic pigments, not only chlorophylls but also carotenoids like carotene, xanthophyll and so on. So in both photosystems there are carotenoid and chlorophyll pigments that are in light harvesting system. If they ask you an MCQ, true or false MCQ, they can ask in light harvesting system there are chlorophyll A and B only. No, that is incorrect. There is also carotene and xanthophyll is there, but in minor proportions. But they present ඒ කියන්නේ එහෙම ඇහුවොත් බෑ chlorophyll A and B විතරයි තියෙන්නේ කියලා කිව්වොත් that is very much wrong. Okay. So in a light reaction, what happens? The basic thing I have taught you. What? The thing happens to a chlorophyll molecule. The light energy passes. What happens? They are absorbed by these chlorophyll molecules, and what happens to them? They are stimulated by this light energy. After that they are excited and they are electrons at a high energy level. Electrons at a high energy level. So what happens when something goes to a high energy level? They try to release the energy and come back to the normal thing. So this electron releases energy to him, but he also is excited here. He also is excited here. So what happens? These electrons, the energy jumps from here to here, and after that finally to P680 molecule. What happens? All the ones all the chlorophylls and other photosynthetic pigments, what do? All of them, what do? They transmit energy from one chlorophyll, one photosynthetic pigment, and to other pigment, and all of them are collected into this special P680 chlorophyll molecule. What happens? Their electron is also stimulated, excited, and goes to a high energy level. Here comes the primary electron receptor. Here there were no any receptors. Only receptor was this one, P680, because all were excited. But now we have a primary electron receptor. What happens to them? after accepting this electron, is try to release this energy, but not at once. Goes through a series of reactions and then goes to this PS1 photosystem. Goes to this PS1 photosystem. So this intermediate action බලන්න මේතර intermediate action එකක් තියෙනවා. මේ intermediate action එකේ මොකද වෙන්නේ බලන්න. In this intermediate action what happens? Energy is released a bit. ඔක්කොම නෙමෙයි, but little energy is released. We call this process photophosphorylation. We call this process photophosphorylation. Photophosphorylation. In photophosphorylation, what happens? ATP is released. ATP is released. ATP is released here. So after releasing ATP, this goes to P700, and what happens? In here also the solar energy causes excitation of electrons, and all of them are complain to this special P700 chlorophyll molecule. What does it do? It does also excite. After excitation, what happens? This is also a primary electron receptor. This is also a primary electron receptor. What happens? The electrons are accepted by this primary electron receptor. After that, what happens? His routine is similar, his task is similar, to push through secondary electron receptors in order to release energy. This primary electron receptor is trying to do that. What happens? It tries with him to release his energy. Little by little. What happens? The energy is released. කොහොමද energy release වෙන්නේ? There is a process happens. NADP turns into NADPH. This reaction is stimulated by NADP reductase. දැන් බලන්නකො PS1 photosystem එකේ ඉඳන් මොකද උනේ energy release කර they came releasing their energy. Then after that what happens? These the electrons are now in its normal energy level. So low energy level A. Low energy level A. What happens? This is of no use. මේක ආයෙ වැඩගත් වෙන්නේ නැහැ. Then what happens here? මේ වෙද්දි මොකද වෙලා තියෙන්නේ? What are the results here? ATP was here and NADPH was here. ATP and NADPH. Only this. Look at this. Here what happens when an chlorophyll molecule comes, when the light energy comes and excites the electron in here, excites the electron in this photosystem, light harvesting complex. What happens? The electron is released outwards. Released outwards. It comes back, but it cannot come to the original station. It cannot come back to the original station. But the energy level would be same later. But here what happens? The electron is excited and now it's in the outer energy level. What happens? This guy, this photosynthetic pigment, usually we take all chlorophylls, but there are others also. The chlorophyll become what? Unattached with this electron. What happens? It's like chlorophyll is now not there, only the electron and the chlorophyll ion. Okay, chlorophyll plus ion. But දැන් බලන්න chlorophyll plus ion එකක් තිබ්බහම මොකද වෙන්නේ? ඊළඟ පාර light energy එද්දි මේක stimulate වෙන්න පුළුවන්ද? මේකේ chlorophyll තියෙනවද? No chlorophyll. So what happens? This chlorophyll need to be reformed to this chlorophyll molecule need to be reformed into this neutral chlorophyll molecule. Chlorophyll ion should be transferred into chlorophyll molecule. කොහොමද වෙන්නේ? And that's where the occurrence of water is important. H2O. You need to remember this, huh? H2O. It is broken down into pieces. H plus, electrons, O2. 2, 4, 4. This is the equation. Water molecules are cleaved by enzymes, special enzymes. The enzymes names are not needed. Enzymes, and they are turned into H plus, 4 electrons and oxygen. So what happens to this electron? This electron is no more. These electrons go and waste. But මේ electron ටත් එහෙම නෙවෙයි. මේ එකම electron නෙවෙයි මේ එක දිගටම එන්නේ. හරි. Any electron can come. But the most important thing is these electrons come and join with chlorophyll plus. So what happens? Chlorophyll plus get electron, then they turn back to chlorophyll, and chlorophyll can absorb more light again. Oxygen තමයි. එතකොට අන්තිමට අපේ result එක මොනවද තියෙන්නේ? Oxygen is also there. හරි? ඔක තමයි light reaction එක. හරි. 1 minute දෙන්න චුට්ටක් බලන්න. Okay. Next, the Kelvin cycle. So, what is the fate? We must know that what is the fate? What is the fate of the results of the light reaction? The results we know ATP, NADPH and oxygen. But we must know the fate of these ATP, NADPH and oxygen. And the electrons are either destroyed or consumed. But oxygen are not destroyed. Oxygens are released to the environment via stomata. You know stomata, I think. Stomata are small holes in the underside of the plant leaves. So, the result should be oxygen, ATP and NADP. So the next step or the Kelvin cycle, is the cycle or is the process where these results are utilized and a complex glucose molecule is synthesized. That is the Kelvin cycle. The Kelvin cycle, when we are learning about Kelvin cycle, there is an important, very important enzyme we must talk about. I taught this that day also. I will quickly recap. That is the enzyme called Rubisco. As the name suggests, it has a dual action. Ribulose, bisphosphate, carboxylase and oxygenase. Ribulose, bisphosphate, carboxylase, oxygenase. What is the carboxygenase part do and what is the oxygenase part do? Carboxygenase part, what happens? It helps to catch RuBP2. It helps to catch RuBP2 carbon dioxide. It helps. And the oxygenase part helps to catch RuBP2 oxygen. The results are also different. This has a dual action. So, in the enzyme unit, we have learned that enzymes are more large. Enzymes are large than the subjects. This is the enzyme. And this is the place where RuBP and the gases meet. There RuBP is hanging to this enzyme Rubisco. What does they do of carbon dioxide and oxygen? Remember this for an MCQ. Carbon dioxide, both carbon dioxide and oxygen fight for the same reaction site of Rubisco. So what happens? Carbon dioxide and oxygen are competitive inhibitors of each other. Carbon dioxide, if the carbon dioxide bound, carbon dioxide act with RuBP and do a certain cycle, as I would say. But when the oxygen is bound here, what happens? The RuBP goes to a whole different section. Whatever. For the Calvin cycle, in the definition of Calvin cycle, what happens in the Calvin cycle? The first thing would be the carbon fixation. So now for a moment we'll forget this oxygen. Right? We'll forget this oxygen. What happens when bound to RUBP? It forms an unstable C6 molecule. Because why? We know RUBP, I have taught you, is a ribulose in pentose sugar group. So it is a monosaccharide. Ribulose. So what happens? It has 5 carbons and this carbon dioxide, right, 1 carbon. So what does they form? A 6 carbon. But so 6 carbon, but what happens? This is unstable. So okay, 6 carbon, this is unstable, so they quickly form an stable 3 carbon molecules, 2 stable carbon 3 molecules. We call them phosphoglycerate, not just phosphoglycerate, 3 phosphoglycerate. The 3, this number does not relate anyhow to the carbon amount. 3 phosphoglycerate. Okay. How many 3 phosphoglycerate? 3 phosphoglycerate is a 2C. So need to make 2. So what happens? This is the first stable product of photosynthesis. First stable product of photosynthesis. So what happens? This is not keeping like this. Make a කෙලින්ම ගිහිල්ලා glucose වෙන්නේ නැහැ. හරිද? මේ කෙලින්ම ගිහිල්ලා එම් වෙන්නේ නැහැ. There is a pathway. There is a special pathway to this. What happens? මේක chemically කියන්නේ chemistry වලට අනුව නම් කියලා දෙන්න යන්නේ නැහැ. මොකද එහෙම වුණොත් time දෙකයි. 3PGA what happens? Using some of the ATP. Using some, not all, right? Using some of the ATP, what does it do? This 3 phosphoglycerate, what? They turn into 2,3 bis phosphoglycerate. 2,3 bis phosphoglycerate. Is it enough? No. This goes through series of reactions and then form glyceraldehyde 3 phosphate G3P. All are 3 carbon molecules. This is also a 3 carbon molecule. Okay. So this is good. For this reaction, all the NADPH for this equation, all the NADPH that have saved from the light reaction and they turn into Na dP. And also some of the ATP are also participating. Some of the ATP also used. This G3P, what happens to them? They polymerizes and they form a chain of, they polymerizes and they form a chain of reactions, and after that finally they prepare glucose. This is not a cycle yet. What needs to be done? The RUBP must be generated again, regenerated. Because right, RUBP is the catcher of carbon dioxide, but now RUBP are wasted. They have go into these molecules, so on, and what happens to them? They have been now RUBP now must be regenerated. So this happens by G3P. So we call it RUBP regeneration. This is called the carbon fixation. First carbon fixation, then we can say this is a reduction phase. Actually this is the reduction phase. And this is the regeneration of RUBP. Okay, that is the Calvin cycle. Okay, and in an essay examination, if when the teacher asks write down, describe the Calvin cycle, what you need to do? Please draw this, draw the diagram, and then write as paragraph. So star mark, so anything. I think paragraph are better in exam, but I did otherwise. So now okay, if you have any doubt, please tell me, so I can look at the chat and I can look at chat and I'll release your, I'll answer your questions. So the NADPH is all NADPH is consumed. All ATP is also consumed. For what? All ATP consumed. The rest of the ATP is consumed to regenerate RUBP. So, and also this is reduction phase. Okay, this is we call the Calvin cycle. Okay. Now look at within a minute or so. ඕකේ. හරි. මම දැන් බෝඩ් එක ක්ලීන් කරන්නම්. මම ක්ලීන් කරද්දී සාමාන්‍යයෙන් රිවයිස් කර ක්ලීන් කරන්නේ. ස්පීඩ් රිකවරි එකක් දාගන්න පුළුවන්, ස්පීඩ් රිවිෂන් එකක් කරගන්න එතකොට. හරි. උඹට තාම මේක අර ලයිටින් ඉෂුව එක තාම ටිකක් තියෙනවා. මම චුට්ට ඇඩ්ජස්ට් කළා කලින් තරම් ඉන්න නැතුව ඇති බට් මම බලන්නම්. පොඩි වක් හරි ගන්න. හරි ඕකේ. ඕකේ. ලයිට් රියැක්ෂන් එක ඉවරලා මම වේගෙන් කියාගෙන යන්න. හරි. සිංහල පාවිච්චි කරලා. මොකද සරලවම තේරුම් ගන්න. දැන් මම මේ කරන එක තමයි ඔයාලා මේ එක්සෑම් එකට කිට්ටු වෙලා. එහෙම නැත්නම් ඔයා එකපාරක් පාඩම් කරපු එක ආයි මතක තියාගන්න ඕන නම් ඔයා කරන්න ඕනේ මේක. හරි. ඔයා කොහේ හරි ඩයග්‍රෑම් එක අල්ලනවා. එහෙම නැත්නම් දැන් ඔයා ඇඳ ඩයග්‍රෑම් තියෙනවානේ. ටියුට් එකේ තියෙන ඩයග්‍රෑම් එක ස්පෙෂල් ඩයග්‍රෑම්ස් ටික අදිනවා. ඇඳලා පස්සේ මම ස්පෙෂල් ඩයග්‍රෑම්ස් අදින්න දුන්නා. එතකොට ස්පෙෂල් ඩයග්‍රෑම් එක ඇඳලා ඔයා ඒක මේ විදිහට ඉක්මනට රිවයිස් කරනවා. රිවයිස් කරද්දී ඔයා පාවිච්චි කරන වොකැබියුලරි එක ගැන සෑහෙන සෑහෙන කල්පනා කරන්න ඕනේ. මොකද නිකන් රිවයිස් කරනවා කියලා හරි. ඔක්සිජන් තියෙනවා, ATP තියෙනවා, NADPH තියෙනවා. ඔව් ලයිට් රියැක්ෂන් එකෙන් තමයි එහෙම හරියන්නේ නැහැ. හරි. වොකැබියුලරි එක හොඳ ස්ට්‍රක්චර් එකකට හදන්න ඕනේ. මොකද මේ රිවයිස් කරන වෙලාව ඉතාල වැදගත්. මම මේ රිවයිස් කරන එකේදී මම ඔයාලා සිලබස් එකේ නැති පොයින්ට්ස් නම් කියන්නේ. එහෙම නැත්නම් අත්‍යවශ්‍යම පොයින්ට්ස් නෙමෙයි නම් කියන්නේ. අතන වැඩක් නැහැ රිවිෂන් එකේ. දැන් කරන්න ඕන රිවයිස් එකේදී ඔයාලා වොකැබියුලරි එක මේක ඩිස්ක්‍රයිබ් කරනකොට ඔයා තනියම Tamanට කියනකොට කියන වොකැබියුලරි එක හොඳට ඔළුවට දාගන්න. ඒක Tamanගේ මේකක් හදාගන්න. ගයිඩ් එකෙන් පිට නොයන්න. හරි. මොකද රුබිස්කෝ වල මේ තියෙන අල්ට්‍රා ස්ට්‍රක්චර්ස් ඕක වැඩක් නැහැ අපිට මේ වෙලාව. හරි. එහෙම නැත්නම් මේ G3P වලින් ග්ලිසරාල්ඩිහයිඩ් 3 ෆොස්ෆේට් වලින් ග්ලූකෝස් හැදෙන මෙකැනිසම් එක අපිට වැඩක් නැහැ. හරි. අපිට we just need the basic thing. Okay? Okay. So, listen to me. So, there were තුනක් තිබ්බා. Products of light reaction. Oxygen, what happened to oxygen? Oxygen went to enter to the environment via stomata. Right? A point is so, and the ATP and NADPH are released into the matrix. Matrix, ne? Stromal, ne? So this ATP and NADPH, the fate of ATP and NADPH is determined by the Calvin cycle. The important thing is the enzyme Rubisco. It has a dual action to bind carbon dioxide and oxygen also. And as they act as competitive inhibitors to each other, carbon dioxide to oxygen, oxygen to carbon dioxide. So what happens? When carbon dioxide comes, carbon dioxide binds with this Rubisco, RUBP plus carbon dioxide which has a single carbon, and this has which is an pentose sugar, ribulose the actual name, has 5 carbon. What happens? They combine and make 6 carbon unstable 6 carbon molecule, but it is divided into 3 phosphoglycerate molecules. 3 phosphoglycerate molecules. So RUBP is like that. So what happens? This 3 phosphoglycerate is the first stable product of photosynthesis, but it does not like that, and it turned into 2,3 bisphosphoglycerate, and then they turn into glyceraldehyde 3-phosphate. This whole process we call it reduction phase. Make chemically reduce වෙන්නේ කොහොමද කියන එක ගැන idea එකක් ගන්න පුළුවන්, but deeply අතරම අවශ්‍ය වෙන්නේ නැහැ. මම ඕන වෙලාවට MCQ වලදී ඕන එකට එතකොට කියන්න. හරි? reduction phase. To this reduction phase, to transfer 3 phosphoglycerate into 2,3 bisphosphoglycerate, some amount of ATP is needed. And to convert 2,3 BPG to G3P, glyceraldehyde 3-phosphate. All the NADPH was used and the some amount of ATP is also used. We call that reduction phase. And from G3P, glucose is synthesized. After that, the rest of the ATP is helping to the regeneration of RUBP. Regeneration of RUBP. So after that, what happens? Carbon fixation occurs again. Carbon fixation occurs again. So that is how we could revise photosynthesis in a nutshell. So I told you that when RUBP binds with carbon dioxide, a special mechanism called the Calvin cycle occurs. But if RUBP binds with oxygen, why is that? Because the Rubisco enzyme has a dual action. So in this dual action, carbon dioxide and oxygen both bind to the same active site alternatively. They act as competitive inhibitors to each other. So there is a 50% chance of binding of an oxygen molecule to Rubisco and react with RUBP, and there is a 50% chance of binding of one carbon dioxide molecule to Rubisco and then react with RUBP. So this is oxygen, this is carbon dioxide. Okay. Meanwhile, you can look at the tube I have sent you. There I have inserted diagrams from the international books as well. Other than this, so oxygen. Now carbon dioxide, story of carbon dioxide is finished. What happens to the oxygen? When oxygen binds to this active site of Rubisco, remember RUBP is already bind to here. RUBP is already bind to here. So oxygen reacts with RUBP. RUBP is 5 carbon, but oxygen does not have carbon. So the result should have only 5 carbon. Look at this. The results are not a 5 carbon stable or unstable molecule. The results are 2. There are 2 results from this reaction. The results would be 1 3 PGA molecule. 3 means not 3 carbons, not 3 PGA molecules. Actually, 3 phosphoglycerate means phosphoglycerate කියන එක meaning එක චුට්ටක් කියන්න. හරි? ඒක that is not important, but look at නිකන් ඒක confuse වෙනවා. phosphoglycerate කියලා කියන්නේ carbon 3ක් තියෙන මේ molecule එකේ අපි මේ molecule එක 1 2 3 කියලා මෙහෙම නම් කරාට පස්සේ මේකේ third carbon ට phosphate එකක් third carbon ට phosphate එකක් bind වෙන එකට අපි කියනවා third carbon ට phosphate එකක් bind වෙන එකට අපි කියනවා 3 phosphoglycerate කියලා. ඒක carbon 3ක් තියෙනවා කියන එකක් නෙමෙයි. Okay. 3 phosphoglycerate, but 1 3 phosphoglycerate. හරි? දැන් එතන carbon 3ක් තමයි තිබ්බේ. but not මේක හින්දා නෙමෙයි. and 2 carbon organic molecule. හරි? 2 carbon 3ක් තියෙන molecule එකක් හැදෙනවා. The name of that is phosphoglycolate. The name of this is phosphoglycolate. Okay. Phosphoglycolate. දැන් බලන්න phosphoglycolate වලට මොකද වෙන්නේ කියලා. phosphoglycolate. Oxygen bind වුණා visco නිසා RUBP එක එතනින් හැදුණා දෙකක්. බලන්න. මෙතන 3 carbon මෙතන 2 carbon. මේකේ අපිට මේ numeric code එක ඒවා මොකුත් අපිට අවශ්‍ය නැහැ. But name you know දැනගන්න චුට්ටක් phosphoglycolate. හරි. What is the difference? Then RUBP carbon dioxide එක fix carbon dioxide fix කළාම what happens? There were 2 PGA molecules. Right? PGA molecules දෙකක් තිබ්බා. ඔක්කොම හැදිලා glucose amount එකක් හැදුණා. දැන් මොකද වෙන්නේ? There's only one PGA molecule. PGA molecule එකට මොකද වෙන්නේ? මේක අර කියන process එක යනවා. That is not the Kelvin cycle. අපිට කියන්න බෑ ඒක Kelvin cycle එකකට යනවා කියලා. හරි? Kelvin cycle එකකට යන්න බෑ. But මේක වෙන්නේ මේ PGA ආයෙ ගිහිල්ලා අර 23BPG G3P and then polymerize and become glucose. Oh matter. Right? But earlier there were 2, but here only 1. Right? Hi. And the fate එක හරි. What happens to this fellow? මෙයා මෙතන react කරන්නේ නැහැ. මෙයා මෙතන react කරන්නේ නැතුව මෙයා මොකද කරන්නේ? This 2 carbon product leaves the chloroplast. This 2 carbon product leaves the chloroplast and this enters into the peroxisome and mitochondria. This goes into the leaves chloroplast and goes to peroxisome and mitochondria. Okay. The mechanism after this is not necessary for you. ඒක තරම්ම තේරුමක් නැහැ. සිලබස් එකේ කොහෙවත් තිබෙන්නේ නැහැ. ඒකේ ප්‍රශ්න අහලවත් ඒක එපා කියලාම තියෙනවා is not necessary කියලා. Okay, what happens? මේකේ නමක් තිබ්බේ නැහැ. But learn. There's phosphoglycolate leaves chloroplast and goes into peroxisome and mitochondria. Then peroxisome and mitochondria, when we were learning about peroxisomes and mitochondria, I think you have learned that regarding the functions, there were in both of them there are the function photorespiration. So photorespiration is this is an half of it, but the other includes the action of peroxisomes and mitochondria. Okay, this is what we call photorespiration. This is what we call photorespiration. Is this good? No, this is not good. This is not good. Why? Only one PGA is produced, so the amount of glucose is synthesized only 50%. only 50%. So, as the net gain in photosynthetic carbon, as the net loss in uh, glucose production is recorded, this is not a good thing actually. So, this is not a good thing. So, the plants. No, plants are not like that. Some plants, actually, many plants are adapted to this condition, adapted to prevent this condition. For that, there is a special mechanism occurs. We call that C4 mechanism. C4 mechanism. Before that, we'll write a bit. Write down the topic photorespiration. Write down the topic photorespiration. Go. Okay. I'll give you some introduction. Write as points. Star mark එකක් දාලා ලියන්න. Write down in photorespiration. In photorespiration, the Rubisco enzyme in photorespiration, the Rubisco enzyme is capable of acting as both a carboxylase and an oxygenase. Both a carboxylase and an oxygenase. Okay, next point. Oxygen and carbon dioxide. Oxygen and carbon dioxide. Oxygen and carbon dioxide act as competitive inhibitors to each other. Next point. RUBP acts catches කියලා sorry acts නෙමෙයි catches oxygen O2 RUBP catches oxygen and synthesize 1 PGA molecule and within brackets දාන්න මේ 1 PGA molecule දාලා within brackets 3C කියලා 3 carbon කියලා කියන්නේ and a 2 carbon molecule within brackets like right phosphoglycolate oh next point PGA forms glucose in the end. PGA forms glucose in the end. PGA forms glucose in the end. Yes, dear. I am recording the meeting. Yeah. This year I am going to record every meeting and then upload to a YouTube channel also. It will be easy for you. So PGA in the end synthesizes glucose. Okay. Next point. This is 50% loss. This is 50% loss in net productivity of glucose. This is 50% loss in net productivity of glucose. Loss in net productivity of glucose. Next point. 2 carbon 2C molecule leaves 2C molecule leaves 2C molecule leaves chloroplast. 2C molecule leaves chloroplast and enters peroxisome and mitochondria. Okay. Draw this down. I will give you 3 minutes. Okay. I will revise while cleaning. Okay. This is the process of photorespiration. Those are the points. They will give you a short note to write in a term text. Robisco enzyme has two parts. Dual action, two actions. Can bind with carbon dioxide and also oxygen. When oxygen binds to Rubisco, what happens? RUBP, as it is a 5 carbon molecule, this both of this combined and produces 3 phosphoglycerate, 3 phosphoglycerate molecule which has 3 carbons, and a 2 carbon molecule which is phosphoglycolate. The phosphoglycerate molecule, 3 phosphoglycerate molecule goes to the same pathway 2,3-DPG, G3P and glucose, but only 50% of harvest is the outcome. But the 2 carbon phosphoglycolate leaves chloroplast and enter to the peroxisomes and mitochondria. That is photorespiration. So it is not a good thing. The photorespiration is not a good thing. So we need to do Photorespiration is not a good thing, so we need to overcome. In the evolution of plants, there are several mechanisms which has been evaluated, synthesized in order to overcome this hazard. One mechanism is the one thing that I am going to teach now. There are several mechanisms in different plants, but the most famous thing is this, the C4 pathway. C4 pathway. Okay. The C4 pathway. Name the name. We talked about the normal photosynthesis pathway, light reaction, Calvin cycle, and in the photorespiration process. Now there's a different photosynthesis process. Previously, until now, all the things occurred in a single cell. Single cell, a single chloroplast. But now here, the mechanism is different. Okay, this we need two cells, and those cells are two types, and those are changed. Those are not similar cells. Those are pretty much different from one another when considering about the photosynthesis process. So, okay, one cell, we call it the leaf mesophyll cells. Leaf mesophyll cells. That's it. Leaf mesophyll cells. And the next type of cell, we call it bundle sheath cells. Bundle sheath cells. Leaf mesophyll cells, and other one is bundle sheath cells. Leaf mesophyll cells and the bundle sheath cells. Okay, I told you two types of cells. Okay, අපි දැන් මේ වෙලාවෙම මේ මතක තියාගන්න. හරිද? මොකද මේකේ logical මොකක්වත් basis එකක් නැහැ නේ. Two types, but okay, they are connected with one another. Okay, they are connected with one another. Yeah, then this is a leaf mesophyll cell, this is a bundle sheath cell. බලන්න මොකද වෙන්නේ කියලා. හරිද? Okay, leaf mesophyll cells, bundle sheath cells. ඔන්න මම දැන් මේ ස්ටෝරි එක වගේ කියනවා මොකද්ද වෙන්නේ කියලා. හරිද? Let's see. Okay. ඔන්න වෙන්නේ මෙහෙම. හරිද? C4 cells. C for plants. Okay, C for plants. We call it C for plants. I'll tell you why is that later after this. In these plants, in leaf mesophyll cells, what happens? Photosynthesis starts in leaf mesophyll cells. In these mesophyll cells, what happens? Photosynthesis starts from which step? Light reaction. Okay. Look at this. Light reaction. Normal light reaction. Okay, so in light reaction, what are the results? What are the outcomes? Lighting in the eye balance. Talking. Okay, light reaction. In light reaction, what are the outcomes? We know oxygen, ATP, NADPH. Okay, we know that these are the outcomes. This happens, then now this is special. Okay, in leaf mesophyll cells, now we are going to start the second step, which is the dark reaction. Why it is called dark reaction? It is not dependent on light. Light independent reaction. What happens? Here we need to, what happens? H2O, from H2O oxygen was synthesized. But now we need to fix carbon dioxide. We need to fix carbon dioxide. For that, what does we do? What can we do? Just wait. Okay, in dark reaction or light independent reaction, now we need to fix carbon dioxide. Fix carbon dioxide. For that, a special molecule called phosphoenol pyruvate. Okay, phosphoenol pyruvate. Phosphoenol pyruvate. We call it PEP. You can use PEP in exam also. You don't need to write phosphoenol pyruvate. You don't need to write phosphoenol pyruvate. This is sheet type. Okay. Phosphoenol pyruvate. So, in phosphoenol pyruvate is a 3-carbon molecule. That is the first change. It has only 3 carbons. It has only 3 carbons. But in RUBP, there were 5 carbons. Here only 3 carbons. What happens? This phosphoenol pyruvate, otherwise PP, is the molecule which fixes carbon dioxide. And as this is 1 carbon, as this is 3 carbon, here he must make a 4 carbon molecule. 4 carbon molecule. So this 4 carbon molecule is another new one, oxaloacetate. This is oxaloacetate. I will draw this nicely. This carbon dioxide is fixed by PEP. And then form oxaloacetate. උඩ චුට්ටක් පේන්න ඒක මම හදන්න දැන්ට කමන්න. PEP oxaloacetate. Okay. So this reaction is stimulated by an enzyme. We call that enzyme PEP carboxylase. Not ribulose bisphosphate carboxylase oxygenase. We call this PEP carboxylase. PEP carboxylase. Hari PEP carboxylase. This enzyme is very useful one. බලන්න. This enzyme did not have any dual action. මේකට කිසි dual action එකක් තිබ්බේ නැහැ. So straightforward. PEP transformed into oxaloacetate, which has here 3 carbons, now 4 carbons with addition of carbon dioxide. Now what happens? This oxaloacetate is then හරි බලන්න මේ oxaloacetate එයාගේ යාළුවෙක් හරිද එයාගේ වෙන මෙකැනිසම් එකකට ගිහිල්ලා හරි oxaloacetate මොකද වෙන්නේ oxaloacetate වෙන්නේ is converted into malate. Oxaloacetate is converted into malate, but the important thing is no any carbon added, always 4 carbons. Okay, then make carbon dioxide is fixed into PEP and then form oxaloacetate, then malate. And then apita ඕනේ මොකද්ද what we need by any mechanism we need to synthesize glucose. අපිට මේ 4 carbon molecules එහෙම හදාගන්න ඕනද? ඕනේ. To synthesize molecules there should be a Calvin cycle. because we need to synthesize phosphoglycerate, 3 phosphoglycerate. Otherwise, how can on earth can we synthesize glucose? Huh? The precursor of glucose is glyceraldehyde 3 phosphate G3P. So to synthesize G3P, there should be PGA. They need to be transferred converted into 2,3-BPG. So this is not what we want. But look at this. These are synthesized in order to help that mechanism. What happens to malate? On the bad malate malate is transported. Malate is transported to the other cells. Malate diffuses to the bundle sheath cells. Okay, malate is transferred, diffused into these bundle sheath cells. Here, malate in bundle sheath cells. Okay, now add 4 carbons here. Okay. In here, in bundle sheath cells, what happened? Malate releases carbon dioxide. Then podi spark malate releases carbon dioxide, and then when an 4 carbon molecule releases a carbon, it becomes a 3 carbon molecule. It synthesizes molecule pyruvate. 3 carbon pyruvate. Okay, what happens to this pyruvate? And then pyruvate is transferred again to this leaf mesophyll cells. Okay, pyruvate is transferred again to this leaf mesophyll cells. What happens to carbon dioxide? Here there is the RUBP. Here there is the RUBP, and this binds with here there is RUBP binds with carbon dioxide with the help of Rubisco. And then the Calvin cycle occurs, and then form sugar, later form sugar. I'll tell. Pyruvate again converted into, this is 3 carbon, converted into PEP with the help of ATP. With the help of ATP. Okay, now what is the importance here in leaf mesophyll cells, here in leaf mesophyll cells, the production of the light reaction were oxygen, NADPH and ATP. If there were Rubisco here, what happens? As usually, they are competitive inhibitors to each other. ඒගොල්ලෝ මොකද කරන්නේ? ෆයිට් කරනවා එකම ඇක්ටිව් සයිට් එකට. එතකොට Rubisco වලින් හැදෙන harvest එක අඩුයි. Here what happens? This PEP carboxylase, PEP carboxylase does not have a dual action. So what happens? Does not have a dual action. So what happens? This is straightforward converted to intermediate molecules. Carbon dioxide waste උනාද? නෑ. Carbon dioxide ඔක්කොම malate වලට ආව one carbon. ඒ carbon dioxide release කරා කොහෙටද? bundle sheath cells. Pyruvate අනිත් පැත්තට ගියා. Release කරපු carbon dioxide, so what happens? Here there is RUBP. දැන් ඔන්න question එකක් එනවා. Here there is RUBP and Rubisco enzyme. So aren't there oxygen? That's a good question. And there are not much oxygen here in bundle sheath cells. Oxygen කොහොමද එන්නේ synthesize වෙන්න ඕනේ oxygen. කොහොමද oxygen synthesize වෙන්නේ? Oxygen are synthesized in the light reaction. These bundle sheath cells are adapted. These bundle sheath cells are adapted to eliminate the effect of light reaction. So light reaction would not happen in bundle sheath cells. So when light reaction does not happen, there is no oxygen. There is little, but not a significant amount. There is no oxygen not much oxygen in this bundle sheath cells. So carbon dioxide can freely bind to this Rubisco, to RUBP via Rubisco enzyme. Here only the light reaction is done, not the dark reaction, not the Calvin cycle. We know that. If the Calvin cycle also was here, what would happen? The same thing. Our Rubisco enzyme has dual action. But එතකොට අර carbon dioxide 100% ම මොකද වෙන්නේ fix වුණා. මෙතනින් fix වෙලා oxygen නැති environment එකකට ඒගොල්ලන්ව transport කළා. Transport කළාට පස්සේ මොකද උනේ? This carbon dioxide are fixed and prepared. Make synthesize sugar. ඊට පස්සේ bundle sheath cell වල Calvin cycle එක වුණා. මෙතෙන්ට යනකොට මොකද උනේ? ආයි pyruvate synthesize වෙලා උනේ. එතකොට this is the C4 mechanism. This is the C4 pathway and C4 mechanism. So what would be the change in these between these two cells? Here light reaction happens. Here Calvin cycle happens. Here Calvin cycle does not happen very much. Very වෙන්නේ නැහැ. Here light reaction එක වෙන්නේ නැහැ. Not very much. So there are several things that we need to identify the differences, key differences between these two cells. So before that, I'm telling you, let's write the note. Okay, I'll I think keep some space. No, we have some time, eh? So I think you make it clear the done. We have a device. Okay. Okay, first write down C4 plants or C4 pathway, both are same. C4 plants, write down C4 plants. C4 plants. මෙතන කැපිලා තියෙන්නේ PP and 3C කියන එක. So then wrote this down. Wrote it down. I will give you some time. If you have any doubt, please ask me or just put a message into the chat. Okay. I'll give you another minute. Okay. Write down points immediately. 'To overcome, to overcome, to overcome photorespiration, to overcome photorespiration, to overcome photorespiration. To overcome photorespiration, and to face, to grow under, to grow under, to grow under, to grow under high temperature, high temperature, dryness, high temperature, dryness, high temperature, dryness, and high light intensities, and high light, high, not highlight, high light intensities C4 mechanism was evolved. මං කිව්වනේ C4 mechanism was evolved කියන එක. එහෙනම් අතනින් ඉවරයි. To overcome photorespiration and to grow under high temperature, dryness and high light intensities, and write down low carbon dioxide concentrations, low carbon dioxide concentrations, low carbon dioxide concentrations. Okay. Write down C4 pathway. Next point, C4 pathway starts in leaf mesophyll cells. C4 pathway starts in leaf mesophyll cells. Leaf mesophyll cells. Leaf mesophyll cells. Next point, carbon dioxide is carbon dioxide is initially fixed. Carbon dioxide is initially fixed with PEP, phosphoenol pyruvate. PEP within brackets write phosphoenol pyruvate. Using PEP carboxylase. As carbon dioxide acceptor. Using PEP carboxylase as carbon dioxide acceptor. Using PEP carboxylase as carbon dioxide acceptor. Right. That's it. Then next point, result is අර PEP කිව්ව තැන පොඩ්ඩක් උඩට මේකක් දාලා ලියාගන්න, කාකපාදයක් දාලා ලියන්න 3 carbons කියලා 3C කියලා ලියාගන්න. Result is oxaloacetate. OAA. Next point, OAA is rapidly converted. Oxaloacetate is rapidly converted to more stable. OAA is rapidly converted to more stable malate within brackets 4C, 4 carbons. Next point, malate diffuses to bundle sheath cells. Malate diffuses to bundle sheath cells. And malate diffuses into bundle sheath cells and bundle sheath cells and converted to pyruvate releasing carbon dioxide, releasing CO2. Here, next point, here CO2 is fixed again by Rubisco then go for Calvin cycle. Okay, next point. Pyruvate transfers back into mesophyll cells, leaf mesophyll cells, and regenerates PEP using ATP. Regenerates PEP using ATP. Okay. Point out. If we want to provide the efficient function, if these two types of cells, leaf mesophyll cells and bundle sheath cells, if they want to provide an efficient functioning, what do they need to have? There should be, this include two cells, two types of chlorophylls, two types of chloroplasts. What they want to have is specific connections, high connections of each other cells. So these leaf mesophyll cells and bundle sheath cells are interconnected with many number of plasmodesmata, high numbers of plasmodesmata. You know about plasmodesmata. So uh, yeah, write down that also. Next point, for the efficient functioning and also the bundle shield cells තියෙනවා නිකන් enclose වෙන ඒගොල්ලන්ගේ vascular bundles හරිද ඒ කියන්නේ මෙහෙම වෙන්නේ හරි ඒ කියන්නේ bundle shield cells තියෙනවා මැද්දේ පිටින් තියෙනවා mesophyll cells පිටින් තියෙනවා mesophyll cells එතකොට leaf එකේ තියෙන cells තියෙන anatomy එක හරි වෙනස් බලන්න light reaction එක වෙන්න light එන්නේ නැහැ ඇතුලේ bundle shield cells වලට ඒවා cover වෙලා තියෙන්නේ mesophyll cells වලින් හරි that leaf structure we call that Kranz structure, Kranz anatomical structure we call that. Okay, write down those points. Okay, for the efficient functioning of C4 pathway, for the efficient functioning of C4 pathway, mesophyll and bundle sheath cells are tightly interconnected. Mesophyll and bundle sheath cells are tightly interconnected to each other by high numbers of plasmodesmata. Plasmodesmata. Next point. The bundle sheath cells enclose the vascular bundles. plants කරද්දී කරන්නේ vascular bundles කියන්නේ vascular tissues xylem and phloem ඔය ටික මෙහෙ මැද්දේ තියෙනවා වට ඒක වට කරගෙන තියෙනවා bundle sheath cells ඒ ඔක්කොම enclose වෙලා තියෙනවා mesophyll cells වල the bundle sheath cells enclose the vascular bundles මොකද vascular bundles තියෙන්නේ the synthesized glucose they are converted into sucrose and passed down to the storage areas via the phloem vascular tissue so in the vascular bundle එක තියෙන්න ඕනේ ඒක ළඟම the bundle sheath cells enclose the vascular bundles and are themselves surrounded by the mesophyll cells and this type of leaf anatomy and this type of leaf anatomy was termed Kranz, Kranz, Kranz anatomy. Okay. Next point, last point. PEP carboxylase enzyme. PEP carboxylase enzyme. PEP carboxylase enzyme is much more efficient. Much more efficient for carbon dioxide fixation. Much more efficient for carbon dioxide fixation than Rubisco. Much more efficient for carbon dioxide fixation than Rubisco. It has no affinity. For oxygen. Okay, now I am going to clean this while revision. In mesophils, leaf mesophyll cells, light reaction occurs. The resultant of oxygen, ATP, NADPH, and in mesophyll cells, carbon dioxide is fixed first by PEP with the help of PEP carboxylase, which acts as the first carbon dioxide acceptor, then converted to oxaloacetate, which has 4 carbons, and then converted into malate, which also has first 4 carbons and which is more stable than oxaloacetate. Then this malate is diffused මේ term එක ඕනේ diffuse කියන එක නිකන් transported නෙමෙයි diffuse, that's the technical term. Diffused into the bundle sheath cells, and these in bundle sheath cells it releases carbon dioxide and make pyruvate which has 3 carbons. The carbon dioxides are then refixed by RUBP with the help of Rubisco, and then goes into the Calvin cycle and make sugar. The pyruvate goes again back to the mesophyll cells, and then pyruvate is again with the help of using ATP, PEP, phosphoenol pyruvate is regenerated. So there are different changes in the mesophyll between the mesophyll cells and bundle sheath cells, but in order to efficient functioning between these cells, there are high number of plasmodesmata, tight junctions with plasmodesmata as interconnections of between these two cells. So the first carbon dioxide receptor in C4 pathway is PEP, phosphoenol pyruvate. This happens in mesophyll cells, and in bundle sheath cells RUBP is the one. So what happens? So the last part we need to know about we need to compare C3 and C4 plants. Now, what is C3 and what is C4? C3 is the definition, when the first stable product of photosynthesis is 3 carbon molecule, we call that plant C3 plant. When the first stable, remember, stable molecule of photosynthesis is carbon 3 molecule, we call it C3 plant. Like the C3 molecule is PGA. When the first stable uh, molecule of photosynthesis is a carbon 4 molecule, 4 carbon molecule like oxaloacetate OAA, we call it C4 pathway. That's the chain. So what are the differences in here? C3 and C4 plus. C3 and C4 plus. Okay. Now you know. First, carbon dioxide fixation. First, carbon dioxide fixation and then carbon dioxide acceptance. Then carbon dioxide acceptor and then carbon dioxide fixing enzyme. Next one. Next, the first product of carbon dioxide fixation. First, stable product. Then leaf anatomy. Then productivity. Easy, too easy these are. මේවා තමයි යහන්නේ term test එක. Okay. First, carbon dioxide fixator. Carbon dioxide fixation occurs once in C3, but here twice. Carbon dioxide acceptor is first in C3. In C3 what? In C3 RUBP. In C4 there are two. First PEP, then RUBP. 5 carbon. PEP 5 carbon in mesophyll cells. RUBP, sorry, PEP, wrong, very much wrong. PEP 3 carbon, RUBP 5 carbon in bundle sheath cells. Okay, carbon dioxide fixing enzyme, we know this is Rubisco here in C3. In C4 PEP carboxylase in mesophyll cells, Rubisco in bundle sheath cells. First stable product here 3-PGA, not PGA, 3-PGA. It is 3C. Here oxaloacetate, it is 4 carbon. Leaf anatomy, here photosynthesis, there are again bundle sheath cells. Bundle sheath, if the photosynthesis occurs in mesophyll cells, here in C3. If bundle sheath cells are present, they are non-photosynthetic. Again, ඒවා මේ function එකට ඕනේ වෙන්නේ නැහැ. They are not photosynthesis. So leaf anatomy අපිට කියන්න පුළුවන් happens in leaf anatomy. මෙතන ලියන්න නැතුව මම පොඩි ඒක තියෙනවා පොඩි homework එකක් ඒකට. Here leaf anatomy, there is the crans structure, crans anatomy. Productivity, here only once and 50% loss එකක් තියෙනවා නේ. Low. Here productivity is high. Okay. Write down. මේ මං ලියලා තියෙනවට ඒක ලස්සනට ලියාගන්න. Okay, done. Fixation one twice, accept RUBP, PEP, RUBP, fixing peak carboxylate, rubiscoing multi cells. First table product, PPGA, so rate and productivity low and high. Okay. Okay. As you are asking about essay questions and so on, I am going to give you a essay question. කියන්නේ essay question එකක් දෙන්නම් පොඩි. ඒක ලියන්න. එතකොට structure එක අපිට discuss කරන්න පුළුවන්. කොහොමත් අපිට next week එකේ අපි cellular respiration ඉවර කරලා next week එකේම revision paper එකක් දෙන්න. You will get 2 revision papers. The first revision paper includes the basics and it would be a smooth paper, okay, and the next revision paper 2 will be a bit hard නෙමෙයි ඉතින් theory එකම තමයි තියෙන්නේ. The first එක smooth විදියට මම හදුවේ නෑ තාම. මේ week එකේ හදලා ඔයාලට ඒක එවන්නම්. next class එක ඉවර වෙනකොට ඒ කියන්නේ next week එකේදී ඒක එවන්නම්. දෙකම මට කරන්න ඕනේ. එකක් කරලා ඒක ඩිස්කස් කරනවා ඊට පස්සේ අනිත් එක කරලා ඒක ඩිස්කස් කරනවා. ඕකේ. ඔක තමයි මේ අපි දැන් ටර්ම් ටෙස්ට් එකක් ඉස්සරහට එයිනේ. in February I think. ඒකට second unit එක ඉවරයි කියන්නේ අපිට හොඳ papers දෙකක් විතර කරන්න පුළුවන්. ඊට අමතරව මේ in addition to these the papers and rapid revision test මම හෙට එවන්නම් photosynthesis වල rapid revision test එක. ඒකේ මර අන්තිමට අර තියෙනවා factors affecting මේ photosynthesis කියලා චූටි part එකක්. ඒකත් MCQ එකකට දාන්නම්. ඒක ලොකු part එකක් නෙමෙයි. මම explain කරන්නම් MCQ එකත් එක්ක. මේ දැන් second unit එකත් ඉවර වෙනවා කියලා කියන්නේ දැන් ඕගොල්ලෝ හොඳ තමන්ගේ track එකක් පටන් ගන්න ඕනේ. කියන්නේ තමන්ගේ track එක හොඳට වැඩ කරන්න පටන් ගන්න තියෙනවා. Because I always say doing biology, chemistry and physics, doing biostream is one of the best decisions in my case and also in your case also, and your life in my life, I don't know. It was a good decision actually to and also uh, and doing it in English medium, you have made a great decision. You have made a great decision. That not a good decision. That is a great decision, because the world beyond us, world beyond A levels, is totally depend on your English language and English books and so on. So that's why that's why I'm doing English medium classes. I have learned in Sinhala medium in biology. I have learned medicine in my medical faculty in English. I like to teach in English because I love to learn in English. So when we are going to the medical faculty also, we don't use any single word in lectures or anything. In day-to-day life we use, but for most of the things in our, when seniors are teaching us also, we use most of the times English things. So let's practice essay questions. That would be great for you to overcome bio. I think bio is very easy than other two subjects, as you have a resource book and so on. So you just need to relax, calm down and study. Study කියලා කියන්නේ මේ තියෙන එක කටපාඩම් කරනවා කියන එක නෙමෙයි. ඔක තමයි දැන් විශේෂයෙන්ම especially in girls මේ රෙකෝඩින් එකත් මම channel එකට දාන්නම්. Especially girls ඒගොල්ලෝ කරන්නේ කොහොමද කටපාඩම් කරන එක තමයි. හරි? එහෙම නෙමෙයි. So දැන් මම essay question එකක් දීලා ඒක discuss කරන්නකොට කියන ඒ කටපාඩම් කරන එක වැදගත්ද නැද්ද කියලා. Definition should be අනිවාර්ය definitely. හරි? Definitions, but other things there's a way we can remember things. ඒකේ තියෙන highlight කරගන්න පුළුවන් වචන ටිකක් තියෙනවා. That's right. Let's practice that structure. So write down. This is not a long essay question, a small short note type essay question. Write down. Briefly describe the crans anatomy. Briefly describe the crans anatomy. Crans anatomy and briefly describe the crans anatomy and chlorophyll structure. Briefly describe the crans anatomy and chlorophyll structure. No. Sorry, මම question එක මම part දෙකකට කඩන්න නැත්නම් පොඩ්ඩක් දිග වැඩි වෙනවා. Sorry. Briefly describe the crans anatomy. නැත්නම් මෙහෙම දාන්න. Briefly describe the, sorry, I'm extremely sorry. Briefly describe the leaf anatomy, not crans anatomy. That you must mention in the answer. Leaf anatomy. Briefly describe the leaf anatomy of C4 and C3 plants. Briefly describe the leaf anatomy. Briefly describe the leaf anatomy between C3 and C4 plants. Briefly describe the leaf anatomy in C3 and C4 plants. Then the most important thing of the question is with regard to photosynthesis. With regard to photosynthesis. ඔක්කොම ලියන්න කියන්නේ මේකේ. චුට්ටක් ලියන්න තියෙන්නේ. points 10ක් විතර ඇති වුණා. with regard to photosynthesis. That's the first part. With regard to photosynthesis. Then next question. Okay. Part 2. Describe the changes. Describe the differences. Describe the differences between. Describe the differences between. Describe the differences between chlorophylls of mesophyll cells, leaf mesophyll cells, and bundle sheath cells. Describe the differences in chlorophylls between leaf mesophyll cells and bundle sheath cells. Okay. Write answers to those two questions and then send it to me, or put it to the group so others also can see. So okay then. Okay. Now it's enough for today, I think. I'll send you the recording as soon as possible after uploading to it. Then yeah, of course. Then good night.
So should we describe it in point form or
No.
as a paragraph?
Essay questions, no. Essay questions, as I'll teach you in next week how to identify the type of question. In some questions they tell list. Listing is pointing out, but here I told you to briefly describe. So you must write paragraphs. ඔන්න ඔය difference එක මේ identify කරගන්න බැරි වෙන හින්දා මේ ලකුණු සෑහෙන වෙනස් වෙන ඒවා තියෙනවා. Not points. Briefly describe. So you must write a small paragraph.
Okay.
Okay then. Good night.
God bless.
God bless.